يعني why they don't penetrate or enter like the phospholipid, like here in this example, they just enter between the phospholipid bilayer. Why here they need to use a transport protein to pass?
Because the particles.
Very good. The size of the particles matters. Excellent. What else? Yes. Miss, what about the ions? Do you remember the ions? You forgot?
Yes.
We talked about it yesterday. We said the ions, like for example, Na plus, Cl minus, K plus, H plus, all of these ions, they cannot pass through the plasma membrane. They need, so it's through the phospholipid bilayer. For sure, they need transport protein to allow them to pass. So here we agree together that facilitated use transport protein. The transport protein allow only the molecule that have big size or even the ions to cross the cell membrane. Look here. In this diagram, I put two types of protein, the channel protein and the carrier protein. Both, by the way, they act as pathway for molecule to pass. But for sure, there's a few difference between the two types. Look here. Look at the protein here and look at the protein here. The first picture shows the protein changing its shape or remain with the same shape.
Changing its shape.
Maintain its shape. Excellent. Doesn't change its shape. It looks like a tunnel that allow the red molecule to cross and pass. شوفوا the small molecule, the green one, they pass through phospholipid bilayer. But the red, since they're big, or they are ions, sorry, they can cross the protein to move to the other side. Here, the protein didn't change its shape. But if we look at the carrier protein, look, step 1 and step 2. In step 2, we can see the carrier protein taking the molecule, changing its shape based on that shape, the shape of the molecule, and move it in the other side. Look, step 1 and step 2. Did you understand it?
Yes.
Okay, so if I ask about the difference between channel and carrier protein, you should say the following. Channel protein allow only the ions to pass. يعني هيدا red particles for sure it's an ion, Na plus or Cl minus, whatever. It's not, it's only ion. But here in the carrier protein, step 1 and step 2, the red molecule for sure in this example, it's a molecule that is big in size, like for example, the glucose, the cell needed, by the way. It has to enter the cell. But since its size is big, it cannot cross the phospholipid bilayer, so for sure it has to use carrier protein. So this is step 1, step 2. Did you understand it? And here, Mr. Differences, just in case you want to study it. Carrier protein change shape to move specific molecule in or out of the cell. For example, of those molecule, the glucose. Channel protein, they have a tunnel. They don't change their shape. Just the tunnel move the ions in or out of the cell. Example of the ions, Na plus, Cl minus. Now let's talk about osmosis. Okay, so yesterday we introduced also osmosis. We said it's a type of passive. Since the type of passive, yani, we need to say the same movement of particles from high concentration area to low concentration area without energy. But that particle should be what? Water, perfect, excellent. So it's the same as diffusion. Osmosis with diffusion, the same. ما بدهم transport protein يا مس. Just they move the particles from high to low concentration without transport protein, but that particle should be water. يعني الأosmosis بيحكي بس عن الwater. Water moving from this area to that area. Miss, we talked about also this example yesterday. Look, this is a U tube, and a tube shaped U. هيدا التube يا miss, it has a thin membrane here. هيدا الممbrane, هيدا black line, it acts exactly like the plasma membrane. Since it acts like a plasma membrane, that means it moves substance selective. Okay, it selects substance that enter and leave. In this example, هيدول يا miss transparent color, يعني white, it's water. But the blue circles represent the sugar. يعني نحن حاطين, we put sugar in water in this U tube. Okay, but we have two sides, the left side and the right. طلعوا يا miss, look before osmosis. عندنا two sides. This is the left and here is the right. Look at the left side. It has less sugar, right? While the right side, it has more sugar. So during osmosis, what happened exactly is the water should move from left to right or right to left?
Left to right.
Why left to right? ليه يا مس the water should move from left to right?
concentration.
Very good, to balance the concentration. ونحن يا مس, we agree to say that always water follow the higher concentration of solute. Since the solute, in this example, the solute is the sugar, right? The sugar is more concentrated on the right, and it's less concentrated on the left. So for sure the water اللي هو colored white, هذا الوايت color between the particles هو الwater. So for sure the water should follow the higher concentration of solute. يعني when في more solute, the water should cross and pass. Since the solute is more concentrated on the right, اتخيلوا دايما أن الwater should cross from left to right. طيب, if I ask you what is the evidence that water crossed this membrane from left to right, what is the evidence? Prove that. How water passed from left to right and not from right to left?
Because what concentration of solute.
No,
Because the concentration on the right is higher.
not the concentration. You have to say the water level. Look at the water level here. مش عم تشوفوا الارrow moving on my screen? Do you see the arrow moving?
Yeah, but it's lagging.
Oh, okay. So look at the right side here, the level of the water, and look here. And after osmosis, we notice that the level of water rises on the right, but it goes down or move down on the left. طلعوا left, miss. It was high, then it becomes low, then the right becomes low, then change to be high. So the water level on the right side rises because water diffused there. This is the evidence why water moves from left to right. Fine. So again, if I ask you in the exam, can you prove that water moved from left to right? You have to say the evidence water moved from left to right is the level of water drop down from the left and rises from the right. All of this process, by the way, happen in order to require or reach a dynamic equilibrium at the end. يعني تصير the concentration in both sides the same. So also yesterday we introduced the three types of solution. نحن حكينا عنهم just in general, but now we'll talk about them in details. Of course, next week I'll try my best to get time to write notes in the class about this. Just in case you like to write any note, I will open it now. You can copy it. Fine. So three types of solution. To prepare any solution, we should have a solute and solvent. أي solution you want to prepare it at home, you get a solvent and solute. Like for example, the solvent is water, and the solute is sugar. لأن الsolute we talked about this yesterday. It's the substance that dissolves in the solvent in water. So by adding water and sugar together, I prepared a solution. That solution, it can be isotonic, it can be hypotonic, and it can be hypertonic. Focus on the word hyper. Hypo, less. Hyper, a lot. Okay? So hypo, less. Hyper, a lot. Iso, the same. How we'll understand this? Have a look on this example. You can understand the type of solution from this example. شوفوا يا مس. أول شيء, let me talk about hypertonic. First of all, to prepare a hypertonic solution, you have to put a lot of solute. يعني لازم تكون solution concentrated. Hyper concentrated يعني having a lot of solute. Okay? So in this example, I took a cell and I put it in a solution. This is a cell, this is the solution. Particles show the solution exactly. So, the first question I want to ask you, is this solution, don't look at the cell, look at the solution that had the blue and the yellow particles. Can I say it's concentrated? It's too much concentrated?
No.
Why? أول شيء
Outside the cell it is.
Yes, Miss هدول blue color, blue dots, they represent water molecule. While the yellow dot or circle represent the solute. نحن we focus on the solute. We don't look at the water. By looking at the solute, the yellow dot inside the cell or outside the cell. نحن لنعرف to guess the type of solution, we have to look inside the cell and in the solution itself. بنطلع بالsolution مثل نشوف a lot of yellow circle, more than inside the cell. So that means the solution here has a lot of solute. It's concentrated. Can we call it in this case iso, hypo, or hyper?
Hyper.
Hypertonic. So نحن to guess the type of solution, just look in the solution and inside the cell. Look at the solute. Where it's more concentrated. أكيد it's more concentrated in the solution لأنه number of yellow circles in the solution is more than inside the cell. So for sure you should say this is hypertonic solution. But just in case you have hypertonic solution that is concentrated. Okay? We need to see the movement of water and we need to see the change in the cell shape. يعني هذه هي cell. Okay? How the cell change shape and how the water moves. By looking at the cell placed in the solution, قلنا أن the solution is hypertonic. Hyper a lot, يعني much concentrated in solute. Yesterday I wrote something on the board. I wrote this. What definition of that?